Tying together all of these scientific and regulatory issues, we might best understand the larger picture in the context of a case study. Let's consider the AquaAdvantage salmon that has been in the news lately. The AquaAdvantage salmon is an Atlantic salmon expressing the Chinook salmon growth hormone gene. It exhibits a dramatic growth rate enhancement early in life, cutting production time roughly in half. It also exhibits a considerable improvement in feed conversion efficiency. With the prospect of shorter production time, reduced costs, improved efficiency, and profitability, it's not surprising that some salmon culturists are very interested in producing the product. The parent company, AquaBounty, seeks FDA approval for marketing the product. In terms of regulatory history, AquaBounty Technologies initiated discussion with the FDA in 1993. That led to meetings between the company and the Agricultural Biotechnology Research Advisory Committee, an interagency committee organized by the USDA. Under the coordinated framework, the USDA was the lead agency to regulate genetically engineered plants, but at the time there was no clear regulatory path for genetically engineered animals. The company petitioned for regulation under FDA because the rigorous pathway for approval might help address public concerns regarding food from genetically engineered animals. The FDA new animal drug approval route has a defined endpoint. The product is approved or it's not. A formal application for an INAD, an investigative new animal drug permit with the intent to commercialize the AquaAdvantage salmon, was submitted in 1995. It's important to note that the AquaAdvantage salmon application included measures to abate environmental impacts by limiting the product definition. That is, the salmon would be produced at a single facility on Prince Edward Island in Canada and grown out in a freshwater land-based culture facility in Panama. Both locations were FDA inspected and featured redundant physical and geographical confinement measures. Biological confinement also was proposed, including production of all female triploid fish. That is, the fish would be manipulated to have an extra set of chromosomes, rendering them reproductively sterile. Against this background, in September 2010, the FDA held a public meeting of its Veterinary Medicine Advisory Committee, or VMAC, on the AquaAdvantage salmon. A detailed briefing package was made available to the VMAC and to the general public. The information contained in the package included all data and information that the agency evaluated in the application process, as well as the agency's evaluations. I would emphasize that such a hearing is unprecedented in the case of a pending decision for a new animal drug application. That is, that it went forward speaks well of AquaBounty's and FDA's intent to be publicly transparent. In the event that the AquaBounty salmon application receives approval, the briefing package will serve as the basis for the Freedom of Information Act summary that normally accompanies new animal drug approvals. In the case of the AquaAdvantage salmon, the key controversies concern the environmental risk assessment. Now, the risk of harm from genetically engineered animals is the product of the harm given exposure to the hazard, that is, the genetically engineered animal, and the probability of exposure. The probability of exposure in the case of the AquaAdvantage salmon was seen to be extremely small due to effective confinement. Land-based production with physical confinement, reproductive confinement, and high stream temperatures downstream from the proposed production facility in Panama. Bill Muir, a noted expert in risk assessment and an author of this CAST commentary, analyzed the data using the net fitness methodology, noting that no net fitness components were enhanced by transgene expression. Hence, natural selection would be expected to remove the transgene from any established population, suggesting a very low probability of harm resulting from exposure to the AquaAdvantage salmon. So, FDA scientists concluded that food from AquaAdvantage salmon is the safest food from conventional Atlantic salmon. Regarding environmental risk, genetically engineered AquaAdvantage salmon are not expected to have a significant impact on the quality of the human environment in the United States, in foreign nations, or in the global commons. In their deliberations that were held at the public meeting regarding food safety, VMAC found an overall appropriate and large number of tests establishing similarities and equivalence between AquaAdvantage salmon and Atlantic salmon. Regarding environmental safety, VMAC found that although the committee recognized the risk of escape from either facility could never be zero, the multiple barriers to escape at both the PEI and Panama facilities were indeed extensive. After the public meeting, 2 separate letters from senators and members of Congress were sent to the FDA Commissioner identifying perceived problems with the FDA's genetically engineered animal regulatory process. In February 2011, 2 senators from Alaska reintroduced a bill that would ban genetically engineered salmon entirely. The House of Representatives in June 2011 adopted such a measure in an amendment to an FDA budget bill that has yet to go to the Senate. The FDA has not yet made a decision as to whether or not to make a finding of no significant impact regarding the environmental review of the AquaAdvantage salmon, or if it will require the preparation of a full environmental impact statement. To sum all of this up, after 16 years, the decision on the AquaAdvantage salmon is still before us. With this case study as background, the 3 authors of this CAST commentary note implications for the future regulation and of the genetic engineering technology itself. We note that environmental risks should be considered relative to alternatives. For example, the comparative risk of sterile transgenic AquaAdvantage salmon may be less than that of fertile selectively bred Atlantic salmon that now escape and interbreed with wild stocks. Second, the current regulatory process focuses on potential risks associated with genetically engineered animals with little consideration of counterbalancing benefits. With a 15-year regulatory history and no decision yet announced, the uncertainty in the regulatory process has essentially halted commercial and public investment in development of genetically engineered animals for agricultural applications in the United States. I would encourage all those listening to download a free copy of this commentary from the CAST website at www.castscience.org.